Wes, you're on mute.
Oh,
No
there we
worries.
go.
Yep.
Jeez. Okay, great. Again, thank you for the introduction. Thank you for having me. Thank you to all of you taking your time to listen to what I have to say here tonight. So I'm not a vet, I'm not an oncologist, but I am a bioscientist, and I'm an entrepreneur, and I'm going to tell you the story tonight, kind of on how we identified, the idea for LEAH Labs and our work to kind of help our dogs that have cancer. So this here is my Chief Inspiration Officer, Layla. She's a pit bull border collie lab mutt, and she is the smartest dog that I've met, but I'm sure that there's some smarter dogs here in this crowd. So tonight I'm here to talk a little bit about a tough topic, cancer in dogs. We never want to hear it, but of course, if we've had more than one dog in our lives, we've probably been touched by cancer in dogs. And so hopefully with this talk, you come away a little bit inspired on some work that not only we are doing, but some other people are doing as well to help our dogs that have cancer. So the objectives for this talk, some fast facts about dogs and cancer, and kind of my opinion and the way that I see the future of companion animal medicine. An intro to what we call the One Health model and how it guides drug development for dogs and their owners. We're going to talk a little bit about gene editing tonight. So my intention here is to get a little bit uncomfortable, a little bit challenged in the science, but I'm happy to answer some questions, and hopefully I can explain it so it can be understood. Our work to bring a new solution to pets that really has never before been possible before gene editing came along. And then through all of this, really, there's a theme of yet again how dogs are helping humans. And so not only is our company focused on helping dogs, but dogs again can help humans in drug development, and so that's a big thing here tonight as well. So the companion animal market is booming. You guys are all here because you love dogs. You're spending your time in a network of essentially strangers, right, to learn dog science, and so that's one way to know that the companion animal market's booming. But there's over 90 million dogs in the U.S. right now. This is up 30% from the year 2000. There's nearly $100 billion spent on companion animals every year and rising. Over 30 billion spent on veterinary care and rising. These numbers are actually probably higher after the year of COVID-19, which really boosted the pet market growth. The average dog owner spends about $2,000 on their dog yearly. I'm sure that that's the case for me and my dog and my fiance's dog and I. And millennials, my age group, we're the largest pet owning population, and 36% of us are willing to spend over $1,000 on our fur babies, and 10% or 11% of us are willing to spend $10,000 or more to make sure that our fur babies, are healthy and happy. But unfortunately, 6 million dogs will get cancer every single year. A third of dogs in total will die of cancer. Half of dogs age 10 and older will die of cancer. These are huge numbers, and cancer really affects all sizes and all ages of dogs. It's not just really old dogs. It can be dogs as young as 2 or 3, and it's all sizes of dogs as well. So it's not just the really big dogs, though there might be higher prevalence of some cancers in big dogs. There's also cancer in really small dogs as well. So if there's any good news about this, it's that canine cancer models human disease really quite well. And so we've been able to study cancer in dogs and cancer in humans and come to some solutions. So for a number of different cancers, a few are listed here. So melanoma, mast cell tumors, osteosarcoma, B-cell lymphoma. These cancers have been studied for humans and also for dogs, and using preclinical models and testing in test tubes. We've developed some drugs that worked for humans, and now we tried them in dogs, or that first worked in dogs and now have been applied to humans. And so what I'm going to spend most of my time tonight talking about is B-cell lymphoma. So this is a naturally occurring model of non-Hodgkin's lymphoma in humans. And CHOP chemotherapy is a chemotherapy regimen that was developed in the 1970s for humans, and it was repurposed for dogs in the 1990s. And really, care for dogs that have non-Hodgkin's, or care for dogs that have this B-cell lymphoma, has really not gotten any better since the 1990s. And so that's what LEAH Labs is focused on helping right now. But for a minute, I'm going to take a step back and talk about the One Health model a little bit. So the One Health model is this idea that, because dogs and humans share 85% of our DNA, we share everything with our pets. If you think about it, we are exposed to the same sun, the same chemicals in grass and in the world, environmental toxins. We eat some of the same foods, or at least dogs eat some of our foods. We share a lot of the same physiology, and because of this, we get the same diseases. And so this can be called the One Health model, where we all we share a lot of the same thing, and so we can learn from one another. So dogs get over 450 diseases, 360 of which are analogous to human diseases, which is really quite fascinating to think about it. And in thinking about the One Health model, the current way that we develop drugs needs an upgrade, and so the One Health model could potentially help that. And so this idea is, if a drug works in humans, it might in dogs, and vice versa. And so there's been a number of scientific publications thinking about this topic. There's a kind of a complicated title of a talk here, or I'm sorry, of a paper that was actually published by a couple of LEAH Labs co-founders. But the traditional way that we develop drugs is through translational medicine. So we induce disease or transplant tumors into mice or pigs. We treat that disease in mice or pigs using drugs that we want to bring to humans. We're able to cure disease in mice or pigs, or sometimes even primates. But then when we translate that to humans, those drugs don't always work the same. And so what we like to think about is using reverse translational medicine, which is this idea that real life experiences of drugs can actually help the application of drugs in different species. And so in thinking about, how the first chemotherapy to be used in dogs for B-cell lymphoma is CHOP chemotherapy, that came from humans. But you could also think about trialing novel therapies in dogs and then taking that to humans as well. And so all of this is because dogs get spontaneous disease. It's a natural disease state. It's not induced disease like we have to give pigs and mice. So here's another example, outside of cancer, an inflammatory bowel disease, and how scientists are studying dogs and humans for, looking at the One Health model and trying to translate drugs from one another to help dogs and to help humans. Here's another one, which is really fascinating. Pets could be showing signs of Alzheimer's disease and human aging. And so there's also work looking at the gut-brain axis and neurodegeneration in dogs, thinking about how we can help humans. But then, importantly for tonight is cancer. And so here's an excerpt from a paper. I'm just going to read part of it here, but dogs have a much shorter lifespan. So their cancers progress more rapidly, enabling researchers to assess a cancer's progress and the effect of a treatment in a year or two, whereas a human clinical trial might take years or longer. And so this is, just a really great instance of, again, dog being human's best friend, where we can hopefully, use dogs to help us use, treating dogs in natural disease states, trying to help dogs live, can also help human drug development as well. So, in all, and thinking about all of this has really led to a boom in thinking about biotech in the companion animal space. And so to help illustrate that, here's a number of headlines, why dogs now play a big role in human cancer research. This was in Wired, I believe. Why pet health is the focus of a new biotech sector. An experimental cancer treatment cured this dog. Could it work for people? Biotech is going to the dogs and big profits await. Gene therapies for cats and dogs. Penn partnered Scout Bio just raised 20 million to make it a reality. 5 reasons why animal health could be the next big wave in biotech. So in today's headline culture, I'm sure that I've convinced you that how animal health is a really hot topic and a focus of medical innovation. So before I continue, I'm just wondering if there's any questions. You can feel free to drop if you have any questions moving forward. Please jump in if you'd like. But I will continue on. So the story of LEAH Labs really began during my graduate school work when I was studying to be a PhD at Iowa State. We were collaborating with some Mayo Clinic professors, and I was developing gene editing technology. So really not thinking about cancer initially. And so here's where we're going to jump into some deep tech about gene editing. So I would imagine, I can't see your hands unfortunately, but raise your hand if you've heard of CRISPR-Cas9. I would imagine a few of you have, if not most of you. And so CRISPR-Cas9 is this new technology that scientists are using every day in the lab to help study genetics. And so really what this allows us to do, it came from an immune system in bacteria. I would love to give a whole presentation on this just because I think it's really fascinating. But what we can do now is use this tool to target 20 base pairs in the 3 billion base pair genome. We can pick one gene and 20 base pairs of that gene. We can go into that spot in the genome and we can cut that DNA. And so that's really important because after we cut DNA, the DNA has to repair itself. And so cells have evolved to do this quite efficiently. Happens thousands of times per day per cell. Right now, in thousands of your cells, the DNA is broken and it's fixing itself. But CRISPR-Cas9 allows us to specifically cut the DNA. So this is important for a reason I'll talk about here in a minute. But after DNA breaks, it needs to be repaired. And faulty DNA repair is why we can get cancer sometimes. So after the DNA breaks, there's a bunch of different DNA repair pathways that can heal the DNA. And again, I would love to go into some science on this because this is what I spent my PhD on. But what I figured out in my graduate work is that a way to kind of control the way that the DNA is repaired so that we can reprogram cells and get them to do what we want them to. So this is the technology. We call it gene weld for like weld and welding genes. And so, I'm sure we all remember from our intro bio courses in, middle school or high school or maybe college, that A's and T's bind to each other and that G's and C's bind to each other. And so you can think about this as magnets, right? The North and South Pole magnets bind to each other, but just imagine there was like an East and West pole and those magnets bound to each other. And so what I discovered is that we can put DNA into the genome just by providing these North and South Pole magnets that match the genome. And so what this allows us to do is to put cargo DNA into the genome to reprogram cells to get them to do what we want them to. And so in my graduate work, we used fluorescent proteins. So we made fish glow, or, you could think about using this for therapeutic cargo. And so here's some really cool pictures that I took as a graduate student where I made some zebrafish glow. And so really what this showed us is that the gene editing technology was working. I was able to put GFP or RFP, the red fluorescent protein or the green fluorescent protein, specifically into the genome under the control of just a single gene. So if you think about it, a gene that's expressed in your skin probably doesn't need to be expressed in your eyeball or in your liver. And so the same thing is here. So here where you see green, this is a gene that's just expressed in the neural tube. Here, if you see red here, these are genes that are just expressed in the blood vessels. And so what my graduate school lab did is that they used this to study cancer biology in fish and different neural development and different other diseases. And so this was a really cool tool that I developed to help our lab study science and study genetics. So we built this and we patented it, and it allows us to now genetically reprogram cells. So the question is, what can we now do with this? So I'll kind of take a pause here if anyone has any questions on what I just went over, but I'm not seeing any, so that's okay. So we started to think about how we could use this tool to genetically reprogram cells and to get them to do what we want them to. So around this time, a new type of therapy came online in humans, what's called CAR T-cell therapy. And this is really fascinating breakthrough technology that is now able to cure lymphoma in humans. So these patients, these human patients that this therapy works for are literally on their deathbeds. They have no other options. Chemotherapy has failed them. They will die, but CAR T-cell therapy is now able to save them. So this is really, really breakthrough technology. And so we wanted to take this tech and bring it into humans. So I'm going to talk a little bit more about CAR T-cell therapy and how it works. So I've already said that it induces clinical responses in over 80% of patients that have been given it. Up to 50% of them are now considered cured. Again, when they would not have made it. Their cancer was taking over their body, but CAR T-cell therapy is able to save them. So the way that this works is it's actually living cells that are used as therapy. So in humans, you extract blood from the humans, you isolate T cells, which are specific types of T cells, use genetic reprogramming to make CAR T cells, you grow these up, put them back into patients that have cancer, and they specifically kill cancer. It's fascinating. So I'm going to go into a little bit more detail. So what is a CAR T cell? Think about CAR stands for chimeric antigen receptor. So if you think about chimera in Greek mythology, right, the fusion between a man and a horse for the centaur. This is a fusion between parts of a monoclonal antibody and the T cell receptor. So the T cell receptor is what tells T cells to kill other cells, and antibodies bind to different shapes. So here, this antibody, if you can envision it, is ready to bind to a triangle right here. So when you reprogram T cells with CARs, what you're then allowing them to do is to go seek out tumor antigens, which are just different shapes on cancer cells, and kill them specifically. So they recognize cancer like a lock and key and then kill it. So here you can see a CAR. This is kind of, this antibody region is shaped like a little semicircle, and it's ready to bind to a tumor antigen that looks like a circle. So different types of cancers express different tumor antigens. Some could be circles, some could be stars, some could be boxes, you get the idea. And so what we can do now using biotechnology is actually engineer different CARs, different antigen receptors that can bind to different shapes on different types of cancer. And so at LEAH Labs, we're the first company to bring CAR T cells to dogs, and we're focused on just one type of cancer right now. It's called B-cell lymphoma in dogs, and it's non-Hodgkin's lymphoma in humans. This is the exact same cancer type that's now curable in humans. So B-cell lymphoma in dogs kills, unfortunately, 250,000 dogs every single year. And there really isn't that great of therapy. So CHOP chemotherapy is the best therapy out there today. It takes 12 to 16 trips to the vet over 5 months, and it only increases the life of a dog for about 10 to 12 months. But the average cost of this is about $10,000. And so, we think this is why only a fifth of these dogs that get this type of cancer are even treated. It could even be less than this because it's such an expensive chemotherapy for something that just doesn't work very well. So, when we were thinking about this business idea, I was thinking about Layla, my best friend. Why is under a year of life and $10,000 an acceptable treatment for, our family members, our furry family members? And at LEAH Labs, we realized that it didn't have to be, because we had the expertise to now bring this breakthrough technology that works in humans and bring it into dogs. So indeed, that's what we do. We use living immune cells to fight cancer. This is FDA approved treatments in humans. Again, really, really effective at killing cancer. And there's hundreds of FDA clinical trials ongoing for different types of CAR T cell therapy going after different types of cancers. Does anybody have any questions? Saving them for the end.
We have a pretty quiet crowd today, but folks,
okay.
Don't be shy. Feel free to chime in with your questions.
Yeah.
Hey, Wes, this is Mike Hoover.
Hi there.
Really excited about your work here. This is really cool stuff. I'd love to ask you a question a little bit more specifically about your T-cell program. And, is the plan to move more into an allo or an autologous program?
Yeah, good question. So what Michael's asking, so in humans right now, the FDA approved treatments are what we call autologous CAR T cell therapy. And so autologous means that we extract T cells from a cancer patient, we engineer them, we put them right back into that same cancer patient. We wouldn't be able to put those into another cancer patient because they would cause toxicities. But allogenic CAR T cell therapy is where we can actually take a healthy donor, take their T cells, engineer their T cells using a little bit more complicated technology, so that now we can give that person's T cells to many different people. And so that's what's called allogenic. Really, other term for it could be like universal CAR T cell therapy. And so, indeed, at LEAH Labs, we're focused on allogenic CAR T cell therapy. So we think that is the future in human medicine. Certainly, there's clinical trials ongoing. It seems to be going pretty well for humans right now. So we're focused on allogenic CAR T cells for a number of reasons, cost being one of them, but also because of what we think is the future in human medicine.
Awesome, thank you.
Yeah. Great question. The other ones are coming at the end, I'm sure. So yeah, at LEAH Labs, this is just a comparison slide for how we think about this. The standard of care for B-cell lymphoma is CHOP chemotherapy. As I said, it's this really expensive, really burdensome treatment that takes 12 to 16 trips to the vet. Imagine if you're a pet owner who lived 2 hours from the closest vet. This is something that you just wouldn't be able to do even if you could afford it, because maybe you couldn't take time off work, this much. And it really is this non-curative standard of care. So at LEAH Labs, we really do believe that CAR T cell therapy could be one trip to the vet. We're targeting a $5,000 to $7,000 price point for this. And because of our platform, we believe that we can do that. We expect an 80% response rate and 50%, potentially cure rate for our therapy. But these numbers are just extrapolated from human medicine right now. We have yet to treat a dog that actually has cancer, but we are very close. Within the next 4 to 6 months, we should be there. And so indeed, right now, we are in clinical development of our therapy. We've run a target animal safety study. So unfortunately, we had to treat some lab beagles, dogs that were born to be brave for science, so that we can move on with clinical testing. And so we hope that this is the last time we'll ever have to use lab dogs in our testing. But for the IACUC, for the institutional animal approvals needed at the institution that we're treating actual cancer patients in a few months, they required us to do this because literally no one has done this in dogs for the platform that we're trying to develop. So we had to do that. But again, we hope to never have to do that again. So the primary endpoint for that, was that the dogs lived through our therapy, and that there was no graft versus host disease. So graft versus host disease is something you have to worry about when you do this allogenic transplant, where you take cells from one person and give them to many people. And so the dogs were dosed on March 9th. They're alive. They're well. They were happy the whole time. There were no toxicities. And so we're really excited to say, that as a company, we have hit this really important milestone, that we've built, our first product, and that we've gotten it into dogs and shown that it's safe. So, here's how we think about the future for the company and our pipeline for identifying a lead asset, which is, the first product, the product that you want to bring through regulatory trials and USDA licensure. So right now, as a startup, we don't have unlimited funding, so we have to think about, what milestones we can reach to unlock more funding. And so right now we're fully funded for our first pilot study with Iowa State. We actually have a grant from Iowa State University that's going to help pay for this study. And so here later this year, we're targeting August to actually treat patients that have cancer. And then we're, hoping to raise a seed round that's going to help us expand our pilot trials so that we can try a few different regimens, maybe give more cells to some dogs, maybe change the pre-treatments that we need or post-treatments that we need that's going to enhance our CAR T-cell therapy's efficacy. But really, as a company, we're focused on identifying what we call a lead asset by Q2 of 2022 and getting into USDA clinical trials on the way to being on the market for our product in 2024. So as I said, we're really excited that we're just a few months away here from actually being able to treat some dogs that have cancer and actually help dogs with our therapy. So just like, in human medicine, there are regulatory bodies for animal drugs as well. And so, rightfully so, we wouldn't want to just be able to treat any dog with anything, under an unregulated platform that would not be safe or good for dogs. And so we're regulated by the USDA. And so the way that this pathway works is if you come up with a new, hypothesis, a new drug that you want to bring to market, you have to validate it, run a safety trial, and then the USDA will permit you to treat client-owned dogs. And actually, the USDA is getting away from this 3-month safety trial because they realize too that treating healthy dogs is not the same as treating sick dogs. And so this actually is hopefully going away. But at the point where the USDA lets you run a clinical trial, really you're only limited by the efficacy of your therapy. And you also, what's really great is you don't have to treat any dogs with a placebo. So we don't have to give anyone's pet a sugar pill just for the study design. So we give 30 to 50 dogs our therapy. We show that it has a reasonable standard of efficacy, and then the USDA can give us a conditional license while we can begin commercial sales and actually selling our product as a company. So in all, the USDA, routes is about 2 to 3 years for about $5 million to get a therapy to market. When you think about comparing that to the US FDA, it's 5 to 10 years and, well over $1 billion to get a drug to market. So this is, obviously a huge, huge difference for a startup to try to bring a therapy to market. But it also opens up the door where LEAH Labs could really bring a lot of value to the human drug development market as well. So taking a look at the human FDA drug approval process, 5 out of 5,000 drugs, 1 in 1,000 drugs will ever make it to people. 1 in 5 in people get approvals. These are not great odds, and billions of dollars are wasted, and billions of time decades are wasted at this. And so I think that we're building LEAH Labs to really help in the preclinical development phase of this. And so nearly all of this, as I said, is done in mice models. I said earlier in the talk, it's all done in mice models, this preclinical development. The disease isn't natural. The mice are just living in a box, and we actually sometimes transplant tumors into mice so they don't even have natural cancer. And we can cure cancer in mice, but when that doesn't translate to people. So we think that LEAH Labs can really have a lot of value here at this spot for preclinical drug testing. So again, showing the same photo because I think it really illustrates the point nicely for reverse translational medicine. But what I think LEAH Labs is doing is really building a discovery engine for novel cell therapy development. So, using reverse translational medicine where real life experiences of drugs and spontaneous disease can be used to inform clinical development, because we share everything with our dogs. And so we think that this is a really awesome model. And so there are provisions in regulatory code where you can actually treat a dog with an experimental product, so long as you do it the right way. You can treat a dog with an experimental product and then be able to use that as data to inform human medicine. And so what we envision LEAH Labs doing, because pharma needs or dogs need new therapies and pharma needs better models, you can do novel cell therapy modeling in dogs that have cancer. So what we're doing is actually giving dogs a chance, with an experimental product, and then also at the same time, using that data to inform human drug development. And so the way that I view this for LEAH Labs is that there's a lot of dogs out there whose pet owners just can't afford therapy, and so their dog, they're going to be on palliative care while unfortunately, their dog's cancer progresses. And so I think that for me, I would really like for LEAH Labs to be able to find those dog owners that have these different types of cancers, and when we're partnering with pharma for studies, we could actually treat dogs, pet owners' dogs that they would never be able to afford a therapy, and actually be able to give those dogs a chance at life while also, informing human drug development. And so I like to call this innovation at the speed of dog years, because we can do this so much faster than human medicine. And so, the idea is we have this candidate funnel, or we partner with human pharma. We start some investigator-initiated trials, which just means, academics or veterinarians that want to work with us, where we can do naturally occurring disease models and N-of-1 studies in dogs that have cancer or have other diseases, where human pharma can advance those hits, something that works well on into humans, and then LEAH Labs can advance those hits into the USDA towards, building a new product line for us. And so, there are additional CAR T-cell therapy indications that we think about for expanding the market. There's a lot of different cancers in dogs, obviously. So mast cell disease is one. This would be the protein target that we would go after. So what we would engineer are little antibody regions to target these different shapes of these different proteins. Glioblastoma is a big cancer in dogs that actually is a really good model for humans, and glioblastoma in humans is one of the most deadly cancers. So we think that this would be a lot of value for our dogs and their owners. And then osteosarcoma, bone cancer is another really tough one. And so also thinking outside of cancer, there's different autoimmune diseases that we think cell therapies are going to be valuable for. And so we know that the FDA is going to require large animal models for this. And so actually right now, as a company, we're partnering with an academic researcher at an institute that I can't say, but we're partnering with an academic researcher to actually trial a novel cell therapy in dogs for an inflammatory disease, inflammatory bowel disease. So I'm really excited about that, and I can't wait to update everyone about that as well. So I'm thinking about some different long-term goals at LEAH Labs. So I myself am a gene editor, and I think that in the future we have a responsibility to help dogs that we've bred into disease states. So there's a lot of genetic disease that, because of the way that we've purebred dogs, we've actually given them diseases or sensitivities to drugs. And so border collies, have this mutation in one gene where if you give them ivermectin, they can have an acute toxicity and die from it. And so, that is a really great candidate to use gene editing to actually fix that diseased allele, so that in the entire population of border collies, then we can allow.
Wes, I can't hear you. I don't know if anyone else is having the same issue. You just cut out.
Yeah, I lost audio too.
Hello,
Okay.
Can you hear me now?
Yes, we can.
Okay, shoot. What was the last thing you heard?
Dalmatians, was it?
Ivermectin and the allele.
Okay, yeah, thanks. So border collies, have a single mutation in a gene that codes for being able to pump drugs out of their cells. And so this is ivermectin is a really sensitive drug for them. And so, not all border collies yet, but like this, we keep breeding purebred border collies. And so at some point it's going to be hard to, or it is next to impossible to get rid of the population. And so using gene editing, we think that this is something that we can fix in the border collie population. But there's a number of other diseases that we've given dogs by the way that we have bred purebreds. Dalmatians get kidney stones, and Huskies cataracts. Bulldogs have breathing problems. Hip dysplasia, Frenchies, and glioblastoma. beagles, and epilepsy, unfortunately, the list really goes on and on. So using gene editing, we do think that there are ways, that we can fix these. But then also something which I think, may raise some eyebrows, but I think it's something to talk about. We certainly have no plans of this right now, but thinking about in the future, where there are already people cloning their dogs. And I think that there are going to be people who might want to think about designer companions. So thinking about labs that don't shed, that could be something that you could use gene editing to engineer, a lab retriever that doesn't shed. Or cats that don't have FeLD1, which is the allergen. Like I'm allergic to cats, unfortunately. But if you could engineer a cat breed that doesn't have FeLD1, and it would become hypoallergenic in that way, I'm not 100% sure that that's what would happen, of course, but there's a possibility. But there's another, other ideas for designer companion breeds, and so I'm sure that could be a point of conversation here as we move forward. So there are additional cool biotechs out there for dogs. I just kind of wanted to touch on here as I wrap up a little bit. So PhytoCure, you may have heard of. They're a company that's using precision medicine for dogs, and so they that have cancer, they take the cancer tissue and they do genetic testing on it, and then they use the AMDUCA, the Animal Medicinal Drug Use Clarification Act, which actually allows you to use any FDA-approved drug that's FDA approved in humans in dogs. Under really no regulatory burden. And so they're doing this. And so their whole point is to help dogs, of course, but they're also interested in repurposing human FDA approved drugs for new cancers that could also go back and help humans. So again, using dogs, helping dogs, but also helping humans by treating dogs. There's two companies that are using gene therapy for cats and dogs, Rejuvenate Bio and Scout Bio. These two are pretty well funded. They've raised, I think, I don't know, 20 or $40 million between them. And so what these guys are doing are introducing healthy copies of dysfunctional genes directly into diseased tissues in dogs. And so again, not only are they helping dogs, addressing an unmet need, but they are models for human gene therapy for the future. And then, another company that's outside of cancer specifically, but their name is Loyal. They're a longevity focused company. And so thinking about aging in dogs. They're studying drugs that help with longevity to help dogs live longer. But dogs are also a model for longevity studies in humans as well. And so, I think last time I heard these folks were actually recruiting dog participants. I'm not sure exactly what it was. It's a non-invasive test, like send some hair or some saliva or something off to Loyal to help them in their studies as well. So I feel like I breezed through that, but somehow it was 30 minutes. Hopefully through this talk, you've gotten some fast facts about dogs and cancer and the future of companion animal medicine. I talked a little bit about the One Health model. Did a quick crash course on gene editing. I'm happy to talk some more about that. Talked about our work to bring a new solution to pets that's not ever before been possible. And then through all of this, I've tried to touch on this theme of how dogs are yet again helping humans. While we continue to help our dogs, they continue to help us. So, there's a number of people that are involved in a startup, a lot of advisors, a lot of funding sources. We've been very fortunate to raise over $1 million to date. So that's been great. I'd just like to always thank these folks who help me run this company since I'm a first-time founder. So there was another thing that I wanted to talk about, which is kind of interesting. So you could actually help us. We're raising money on WeFunder. So this is an equity crowdfunding site. So this isn't like, so it's sort of like Kickstarter. So you guys have probably all heard of Kickstarter. If you go onto Kickstarter, you can support a company, give them $100, and then you get a deal on a tech gadget. But actually, what WeFunder allows you to do in equity crowdfunding allows you to invest into startups and actually get ownership in the company. So with just $100, you could go on if you believed in LEAH Labs and you kind of thought that this mission was cool, you could actually own part of our company. And maybe you don't have any interest in that, but certainly maybe some of your dog owner friends or people that actually might have an interest in that. So just a little bit of shameless self-promotion about this really unique model of how we're raising money. And so equity crowdfunding allows anyone to invest. It's not just accredited investors. So it really is this new asset class. So I would just encourage you to check out WeFunder in general, because I think it's a really cool idea. You can maybe find some cool companies, instead of investing on Robinhood, you could go and check out WeFunder, maybe find a cool company to back and actually get equity in it as a startup. So here is our profile for that WeFunder. Of course, Layla's the star of the show. And to date on this WeFunder platform, right now we've raised $287,000, which is not small money. So we're doing okay. The last time we raised a WeFunder, this is actually our second one, we raised $480K. So we're now up over $750,000 raised on this platform. So that's really cool. I think it's awesome because it gives dog owners a chance to invest in a company. They made a lot of our investors have lost dogs to cancer, and so they write notes to us in this "What People Say" section that really says how why they invested. And for me, it's really motivating to know that. And so I just wanted to show you guys that if you wanted to share our profile or maybe even think about an investment, we would really appreciate that. So anyway, thank you for listening. Thanks to Sonali for the invites and this really cool community. And I would be happy to take your questions. You can reach me at my email at any time, or you can follow us on Twitter, Facebook, Instagram. We're LEAH Labs Inc. On all the platforms. So thank you.
Awesome. Thank you so much, Wes. That was really fascinating. I have a couple of questions for you because I want to give folks a little bit of time to gather their bearings, and then folks feel free to chime in. But actually I have two questions. One is around, we're talking a lot about funding, right? And you did show us the difference between bringing a human drug to market and then bringing cancer treatment for dogs. Can you tell us what you anticipate to be the cost for a dog owner, from A to Z, for treatments like this in the future? I saw on your timeline 2024 is when you hope to be on the market. And secondly, which I think may be something that a lot of folks are thinking, how does the dog fare during this treatment? What is the process like, both for the owner and the dog? Because we're talking in terms of like editing genes, are we injecting this into the patient? And what have you seen so far in terms of quality of life, and how is that different from chemo? Because chemo can be really, really hard on our bodies. So two-part question.
Yeah, so the first question was about taking a drug from where we're at today to getting it to the market, or actually what it might cost for a pet owner?
Actually both. Yeah, if you can tell us, but what you think the approximate cost will be.
Yeah, for sure. So, as a startup right now, we have to think about not only the first drug that we're bringing to market or working to bring to market, we have to think about building a pipeline of drugs so that if the first one doesn't quite make it to the finish line, we have others coming up, right? And so we also have to think about the different value inflection points along the way so we can fundraise off of, the value inflection points so we can keep moving forward. And so for us, I envision from here out, we hope, we envision that it's probably going to be somewhere between $5 to $7 million that we would need to raise in total to get our therapy to market. So in this round, I'm looking to raise a certain amount of money. You're not necessarily supposed to talk say that number. So we do have a target that we're trying to raise, and then at the next value inflection point, we'll raise probably double that, maybe up to 5 million. I'm not raising that amounts right now, so I think I can say that. But so for us, we do think it's about that $5 to $7 million to get a drug to market, from scratch. And so I showed on that slide, $2 to $3 million to get a drug to market for the USDA. And so I think once we're kind of up and running and we have all the wheels greased, I think that it really would be about $2 to $3 million to have an idea and actually get it through the USDA. So the second question, as far as what it might look like for a pet owner to get this therapy. So we know that around $4,000 or $5,000 is where pet owners really start to make decisions on whether or not they can, that their dog, that they can spend that much on their dog. Of course, some dog owners would spend $100,000 on a treatment, but that's not very many. And so we're targeting a price point of about $5,000. The current standard of care is anywhere from $7,000 to $12,000. If you have some complications from the chemo and you have a hospital stay, it can be up to $15,000. And so, we really are targeting about that $5,000 point so that we can cut the market or cut the standard of care in half and still be able to make money as a company, but also get a therapy to dog owners that's cheaper. So, our first therapy, we like to kind of think of it as the Tesla model. So the Roadster was really expensive, but then they worked their way down to the Model 3. And so thinking about that, our first therapy maybe is going to end up being a little bit more, like 8 to 10,000. So it might be a little bit for the high end dog owner, I guess, if you will, at that point. But certainly as we work to scale, we want to get that down lower. And so functionally what it would look like, again, we haven't treated any pets with cancer, unfortunately. But again, in August or September, we hope to. And so maybe, next year at this time, we can have a follow-up chat and talk about our great progress since then. But, the way that we envision it is, since we're building this allogeneic CAR T cell therapy platform, we actually just have, our CAR T cells are frozen. So that we inject nothing into the dogs except the actual living therapy. So the cells would be frozen, in our freezer. We'd have a vet that qualifies their dog for us as a therapy. We ship our cells to them, and then they would infuse them, thaw them over the course of a couple minutes, and then infuse them into the dog. So in humans, CAR T cell therapy can have some strong toxicities, and so we envision certainly for the first few dogs in our clinical trial, we're going to be hospitalizing them for the first 3 days, probably, to monitor them closely. But, in the future, right now with humans, they're even thinking about now outpatient CAR T cell therapy treatments. And so we envision the same for dogs, that once we learn the toxicity profiles, we're going to be able to manage that in an outpatient way.
Got it. Thank you. We did have a question from Diane, and she wanted to know if the treatment is available, or perhaps she meant will be available in Canada. I'm assuming you were first launching in the US and then you would potentially take it abroad.
Yeah, exactly. So first in the US, I've actually not necessarily looked into the regulations in Canada and how that might work. But I have talked to a couple of vets in Canada who thought that it was interesting. Of course, I think somebody's bringing CAR T cells to dogs, whether it's LEAH Labs, I believe that it will be. But in the future, your dog will be able to get CAR T cell therapy. And so, it's just the next logical progression, I think, of the field of medicine. And so whether it's us or someone develops it in Canada, I do believe that this will be available in Canada at some point.
Great. Folks, if you have questions, feel free to unmute and ask. I do have a list, but also it's sessions for you. So yeah, don't be shy if you have questions.
Hey, Wes, I had another question about thinking back on your pipeline. Looked like you were working primarily in CAR Ts. Is there any plan to move into maybe a TIL or a CAR NK type program, or are you primarily focused on CAR T right now?
Yeah, great question. The TILs, not so much. So these stands for tumor infiltrating lymphocytes. So TILs, we are on that. But CAR NK cells, so my employee, my first employee, first hire, really great guy, his PhD expertise was actually in natural killer cells. And so we certainly have an interest in that. I think that natural killer cells, they're going through trials right now. And so I think that that's certainly, maybe in dogs actually could be better than a T cell program, especially in the allogenic sense. It could be better, could be faster, because, the dog cancer is so much more progressive and deadly in dogs that maybe a CAR NK cell would act faster than a T cell might, and actually be able to reduce or eliminate tumor burden faster. So I would say that is absolutely on our pipeline for a trial. But right now as a startup, we have to stay rather focused and rather lean, and we don't necessarily have a full team quite yet to have someone be working full time on CAR NK cells yet.
Awesome, thank you.
Yeah.
Okay, great. We have about 5 minutes, folks. So anyone with any other questions, feel free to jump in. Wes, another question I have, which is very much a layperson question, in case anyone in the audience was feeling a bit shy about asking this. You mentioned that, you described potentially that folks who can't afford it, can't afford the therapy, would be able to participate in pharma studies. And I was hoping you can expand on that a little bit. So my understanding was that pharma is interested in participating in using dog models for human trials because, we share 85% of our DNA and a lot of overlapping diseases and so forth, physiology, et cetera. And so they're interested in performing this research. And for folks who are unable to afford or want earlier access to these treatments could potentially enroll in those studies. They would receive the same treatment as a dog that is undergoing treatment elsewhere, but they would be able to participate and avail of the treatment. Is that the correct understanding? Am I jumbling that up? Just if you want to expand a little bit.
No, that's exactly right. So, I think about all the dogs out there that don't get the chance to have any sort of therapy because their pet owners just can't afford it, at all, So the most common palliative care regimen is just high dose steroids, really cheap steroids that we give our dogs to help them be comfortable when they have cancer. And so, so I mean, even our dogs, the first trial here at Iowa State, we're going to be recruiting from pet owners who actually get to Iowa State before they really learn a whole lot about what the costs are, but they want the best for their dog. They're taking their dog in to learn about what the potential treatments are for their dog that has B-cell lymphoma. And then they're the ones that are actually not going to treat their dogs. They're just going to give them prednisone. At that point, that's when they're going to be eligible for inclusion in our trial. And so already from the start, we're already setting this precedent that we want to help dog owners who cannot afford treatments. And so where the human pharma partnership idea comes in is the idea that down the line, as we build out LEAH Labs to be a partner for human pharma and trialing novel therapies in dogs that are human drug candidates, is that we would do the same thing. We would try to recruit these pet owners who, aren't able to afford treatments so that, we can help them, help their dogs, give their dogs a chance at life. It is an experimental treatment, so, it may not work. But it would give them a better chance at life than just palliative care at that point. So yeah, that's exactly right.
And are there any age cutoffs for the first set of trials?
Yeah, so I think that we are kind of looking for 5 to 7 years, 4 to 7 year old dogs. So because we are a startup, we want to ensure that the first chance that we have to treat dogs is going to be our best chance. To find a dog, that has the potential to rebound and has the potential to respond to our therapy. So we will have some, what we call inclusion criteria in clinical trials for that. But certainly, unfortunately, probably won't be looking to treat a 12-year-old dog. But hoping to find some dogs that are 4 to 7 or so.
Got it.
But that may also be contingent upon the IACUC and how they approve us or not. Yeah.
Right. That makes sense. Let me take a pause for a second before I move on to my next question, if anyone else has thoughts on this. You also shared a bunch of other companies that are doing research in canine cancer and aging and longevity. I'm curious what you think will happen in the next, there is a boom in we're all adopting pets, whether we're purchasing from a breeder or adopting from a shelter. Where do you think the science will go next? Like, what are the questions that people are going to try to solve for? This might be a philosophical question, or you can give me a personalized take. But the reason I always ask this question from our presenters is because I think it makes me hopeful to think that this type of work is being done to support our pets. So I'm just curious what you think is coming next, since you're already sort of at the leading edge.
Yeah, I mean, I think it's all, this idea of personalized medicine. So how in humans, it's, medicine is becoming very personalized. It has to do with your genome and your genes and how your proteins and your cells are messed up, or however they are, whatever disease you might have. I do think that it's all going to become a very personalized thing. And so further, there's I could have included another company on there called PetDX. They're working on liquid biopsies for dogs, just kind of as a monitoring thing. And so if you could get the cost of a liquid biopsy to be cheap enough that every year when you go to the vet, or every 6 months when you go to the vet, you could pay for a liquid biopsy to look for really early signs of cancer and these just little teeny tiny DNAs that are floating around in your cell or in your blood. If you could look for cancer really, really early, you're going to have a lot better chance at fighting it off. And so unfortunately, by the time a dog is diagnosed with B-cell lymphoma, they're like stage 3 or stage 4, like really, really late. Whereas if, it's caught at stage 1, it's probably going to be curable. Whereas, later stages, that's where you need something as powerful as CAR T cells. So I do think that as, personalized medicine and diagnostics get better and better, it's going to give companies like us, an even better chance at having a therapy that is really, really effective for dogs with cancer. And as I said, I think that, gene editing has the potential to, help dogs that we have bred into really just states of disease because of the way that we want them to look. And so I think that we have a moral obligation to help fix that.
Yes, indeed. That was really well said. Thank you so much again, Wes, for doing this. And thank you to everyone in the audience for joining us. I know some folks will be watching this later, so just a heads up, this is being recorded. And I will be sending out a follow-up email where I'll include links to LEAH Lab and the WeFunder and Wes's info if you have questions. And if you're curious, if you want to do a follow-up, Wes, I'd love to have you come back once your trials are done to hear about the data and hopefully great results. But yeah, thanks everyone for joining us. This is our first session during National Pet Week. We do have 2 more sessions. On Wednesday, we have a biocultural anthropologist who is going to reflect upon the trends that Wes touched upon, which is the rise of pet parenting, mainly in an American context, but she's actually lived and traveled all over the world. So she's written a book on the topic, and she has some really fascinating insights about how we parent our dogs. So if you'd like to, feel free to join for that on Wednesday. But otherwise, thank you so much, everyone. I really appreciate you joining us tonight. Have a good night. Bye.
Thanks, everyone.
Bye.
Thanks, Wes.